Welcome back to the deep dive. We're here to basically cut through the complexity of key research papers, making sure you walk away with the core insights fast. You wanted a shortcut into invasion ecology's foundational ideas. Well, today we're tackling exactly that. We're zeroing in on a really influential 2005 paper. It's by Julie L. Lockwood, Philip Cassey, and Tim Blackburn. And the title is The Role of Propagule Pressure in Explaining Species Invasions. White. And this isn't just, any paper. It really lays out a core theoretical framework for figuring out how non-native species actually manage to get established.
That's absolutely right. And our job today is to really distill their main argument, which is quite powerful. They essentially conclude that propagule pressure, let's call it PP, like they do, is the single most consistent, the most critical factor in determining whether an introduction succeeds or fails. Especially now, with global trade accelerating and species moving around faster than ever because of us, understanding PP is just vital if we want to build better predictive models. It's really about getting ahead of the curve.
Okay, yeah, let's unpack that straight away. Propagule pressure. It sounds, well, kind of intuitive, doesn't it? More tries, more success. But what's the actual formal definition they're using?
Right, it's more specific than just more is better. Formally, propagule pressure, or PP, is a composite measure. It captures the total introduction effort, and it breaks down into two really distinct components. First, there's propagule size. That's the absolute number of individual organisms released in one single event. And second, there's propagule number. That's simply the count of how many separate release events happen. So the core hypothesis is pretty straightforward. As you increase either the size of any single release or the number of times you attempt to release, or both, you increase the overall PP. And that increase in PP is thought to directly boost the chances of that species establishing itself.
Makes sense,
right?
Yeah, it does. And splitting it into size and number seems really important. It underlines this distinction they make that PP is an event-level characteristic. That's a key insight, isn't it? Because it sets PP apart from, well, almost everything else people usually look at. Think about species-level traits, like, say, whether a bird eats lots of different things.
Dietary generalism.
Right. Or location-level traits, like how many native species are already there.
Yeah.
Those things are constant for that species or that place, no matter how many times you try to introduce it. But the PP, the actual effort involved in the introduction, that can change drastically with every single attempt.
Precisely. Take rainbow trout on Coryncus mykiss. They're introduced all over the world, successfully in many places. Now, its success could be down to its traits. It eats lots of things. Or maybe the lake conditions are just right. Those are constant factors. But whether that specific attempt in that specific lake worked or failed often comes down to how many trout were actually put in the water that time, the PP for that event.
I see.
So if you're only looking at the constant traits to the species biology of the location's features, you're completely missing potentially the most crucial variable that decided the fate of that introduction right at the start. You're missing the effort.
And the theory behind why these numbers, size and number, matter so much, it's actually borrowed from a field focused on the opposite problem, isn't it? Conservation biology.
Exactly. It's kind of ironic. Conservation biology spends a lot of time figuring out why small populations fail. And the theory points to two big dangers for small groups. First, there are stochastic effects. Just bad luck, basically. Random environmental swings, like a harsh winter right after introduction, or, just demographic chances, maybe all the first offspring are males. Second, there are inverse density-dependent effects. The most famous one is the alley effect.
Okay, hang on. Let's just make sure we explain the alley effect clearly. That's where below a certain population size or density, the individuals actually do worse, right? Their survival or reproduction rates drop.
That's it, exactly.
Like, they might struggle to find mates, or they can't effectively defend against predators as a group, or maybe cooperative hunting breaks down. So being small is inherently risky.
Precisely the mechanism. So putting in a large number of individuals in one go-high propagule size helps the new population just, muscle through those early random accidents. It buffers them against bad luck and gets them above that dangerous alley effect threshold.
Okay, makes sense.
And then making repeated releases high propagule number works differently. It acts like a continuous reinforcement. Maybe the first release was poorly timed, wrong season, or maybe they landed in a patch of bad habitat. Subsequent releases keep adding immigrants, propping up that struggling population. Plus, if you release them in slightly different spots, spatially disjunct releases, it increases the odds that at least some of them will land somewhere with really favorable conditions. It's like hedging your bets.
What's really striking, looking at the sources they cite in the paper, is how consistent the evidence becomes once people actually start measuring PP. It sounds like for decades this was a huge blind spot because, well, nobody was systematically recording the number of individuals released or the number of release events.
That's true. The data was often missing or just hard to find.
But when researchers did manage to dig up that information, maybe from historical records for bird introductions or fish stocking data, or even by doing controlled experiments, the results seem pretty unanimous.
Yeah, they pulled together a really impressive list. Table 1 of the paper. It covers water striders, insects, fish, birds, mammals, using both observational data from the past and controlled experiments. And the finding just keeps popping up across all these different groups, terrestrial, aquatic. When you can quantify the introduction effort, the PP, it consistently explains a significant chunk of the variation in whether the species is established or not.
That level of consistency is pretty rare in ecology, especially invasion ecology, which can seem so messy and unpredictable sometimes.
It really is.
And this leads to what I think is the most surprising or maybe challenging finding in the paper. It's how PP often seems to almost overwhelm or mask the effects of other factors we thought were important. It swamps them, as you said.
It absolutely does. And this helps explain why invasion outcomes often seem so idiosyncratic, so random. Why does the same species make it big in one place but fail instantly somewhere else, even if the environment looks similar? Well, often it's because the human effort involved in PP was just vastly different between those two introduction events.
Right. And the statistical modeling they reference really drives this home. They talk about a study on non-native birds.
Yes, reference 16.
Okay, so in that study, when the researchers included propagule size in their statistical model alongside other potential factors, only two things came out as significant predictors of establishment success: propagule size itself and being a dietary generalist. That's it.
Right, but here's the kicker, the really telling part. When they ran the same analysis on the same data, but this time they deliberately left out propagule size, suddenly a whole bunch of other species traits started looking important.
Ah,
Things like having a large native range or being non-migratory or originating from mainland rather than an island. These all popped up as statistically significant predictors only when PP was ignored.
wow. So the implication there is huge. It suggests that these other traits, the ones ecologists have focused on for years, might only appear to be important predictors because they happen to be correlated with the introduction effort.
Exactly. It's a confounding effect.
Maybe species with large native ranges just happen to be introduced more often or in larger numbers because they were more available or popular. It wasn't the large range itself causing success, it was the higher PP associated with it.
That's the argument. Those traits were confounded by propagule pressure. So the fact that species traits sometimes seem like weak predictors might just be an artifact of not having the PP data in the model, a statistical omission problem.
Okay, so if sheer numbers PP matter this much, how does this translate into, actually managing invasions? The paper shifts gears a bit here, moving from explanation to application. They bring in this idea of the dose-response curve.
Yeah, this is where the theory really meets practical management questions. Think of it like medicine. You have the dose, the propagule pressure, and the response, the probability the species establishes. The shape of the curve connecting those two tells you a lot about strategy. For instance, if the curve is basically a straight line, or maybe exponential, then every single individual you prevent from being introduced gives you a roughly proportional benefit in reducing establishment risk. Reduce the dose, reduce the response.
Okay.
But what if the curve has a threshold? What if it goes up steeply at first but then flattens out?
Ah, like a saturation point.
Exactly. Beyond a certain propagule size or number, adding even more individuals doesn't really increase the establishment probability much further. If that's the shape, then trying to prevent introductions above that threshold might be, well, ecologically redundant. Your management effort is better spent elsewhere, perhaps focusing on stopping introductions below the threshold. The shape is crucial for allocating limited resources.
Which leads right into that tricky question: size versus number. If you have limited resources for prevention, which aspect of PP is more critical to target?
That's a tough one, and likely context dependent.
We know theoretically that size helps overcome those density-dependent hurdles like the alley effect right at the start. Number helps buffer against bad luck spread out in space or time, like hitting a patch of bad weather or unsuitable ground.
Right. And different management actions target these differently.
They mentioned that ship traffic modeling study by Drake and Lodge looking at aquatic invasions. What did that find?
That was interesting. Their model suggested that focusing on reducing the probability of any given ship successfully causing an invasion, which is more related to reducing the size or density of organisms in ballast water, So it was actually more effective overall.
More effective than what?
More effective at slowing down what they called biotic homogenization than, say, trying to completely shut down traffic from a few key high-risk ports, which is more related to reducing the number of introduction events.
Okay, let's quickly define biotic homogenization. That's the undesirable process where ecosystems around the world start looking more and more similar because the same few successful invaders are spreading everywhere, right?
Precisely. Humans accidentally mixing up the world's biota. And in that specific marine context, the model indicated that reducing the dose per ship size was a better lever than reducing the frequency of ships from certain places.
But that's just one context,
Yeah,
often. Just
absolutely.
like
And the
that.
complexity doesn't stop there. PP also has really interesting interactions with genetics.
Right, the basic idea is simple enough. More individuals introduced, higher PP, means more genetic variation arriving.
Exactly. Which helps the new population avoid bottlenecks, founder effects, genetic drift.
Yeah.
Basically gives it more raw material for adaptation.
But they mention a more advanced, kind of counterintuitive genetic effect, too.
Yes, this is really fascinating. Sometimes increased PP can actually result in the introduced population having higher genetic diversity than any single population back in its native range.
How does that happen?
It happens if the invasion pathway, the ships or the pet trade or whatever, inadvertently scoops up individuals from different genetically distinct populations within the native range.
Ah, so if the species has some natural geographic genetic structure back home?
Right. And the transport process mixes individuals from those different regions together at the introduction site.
They use the example of the brown anole lizard, Anolis sagre, in Florida. Is that what happened there?
Apparently so. The introductions likely brought together lizards with distinct genetic backgrounds, different haplotypes is the technical term, from various parts of its native range, like Cuba and the Bahamas.
So essentially, you're mixing different genetic lineages that might not normally interact much, like blending several locally adapted varieties together in a new place.
Exactly. And that sudden fusion can create novel genetic combinations and significantly boost the overall genetic variation available to the population. This could potentially fuel rapid evolution and help the species adapt and spread much faster than it otherwise would.
So it's not just more variation, it can be novel variation, too.
Potentially, yes. It could help explain why some introduced species seem to undergo rapid evolutionary shifts after arrival.
Okay, so PP is clearly central, but the authors are careful to point out it doesn't operate in a vacuum, right? It interacts with the environment and the species' own traits.
Absolutely. That's the necessary nuance. While PP is this consistent factor, its effect isn't uniform everywhere. The dose-response curve we talked about, its specific shape isn't fixed.
It's context-specific.
Exactly. They note, for example, that locations that are already heavily disturbed may be suffering from pollution or physical damage or fluctuating resources, often require a lower level of PP for an invader to get established.
Ah, so it's easier to invade a system that's already stressed or unstable. Takes less effort, fewer individuals.
Seems that way. And similarly, some species just have life history traits that make them good at establishing from very small numbers. Think weeds that produce tons of seeds, or insects with rapid reproduction.
While others might inherently need a really big push, a substantial PP, to overcome those initial hurdles, regardless of the location.
Right. So the effectiveness of a given level of PP depends on both the species and the specific environmental context it lands in. It's an interaction.
Okay, let's try to bring this all together then. Summarizing this deep dive into Lockwood, Cassey, and Blackburn, they've built this really strong case, theoretically and with evidence, that propagule pressure is probably the single most consistent factor we can point to that correlates with establishment success. And crucially, it helps explain why invasion outcomes often look so unpredictable or, idiosyncratic. A lot of that randomness might just be undocumented variation in introduction effort.
That's the core takeaway. Absolutely. It provides a powerful explanatory framework for understanding past invasions. Why did this species succeed here? Check the PP.
Yeah.
But, and this is a big but, they emphasize there's a critical limitation. While PP has great explanatory power for looking backwards.
Its predictive power for future invasions is much less certain, right?
Exactly. It tells us why things happened, but using it to reliably predict which species will invade next or where is still a major challenge. That predictive ability remains largely untested.
That feels like a really significant gap. So the next big step for the science isn't just finding more historical PP data, is it?
Probably not the only step.
It seems like we need more Carefully designed experiments, studies that deliberately manipulate both propagule size and propagule number, maybe across different levels of environmental disturbance or competition.
Precisely. We need to move beyond just describing the patterns of the past and really start testing these mechanisms prospectively under controlled conditions if we want to build genuinely predictive tools.
From explanation to prediction.
That's the goal. Which brings us to a final thought, something for you, the listener, to consider. If the main forces driving propagule pressure today are fundamentally human activities, things like global trade volumes, shipping routes, horticultural trends, the pet trade, basically economic and social forces,
Then how much can purely ecological interventions focused on managing specific sites or blocking specific pathways really achieve in the long run if the underlying rate of introduction effort, the sheer global scale of PP driven by human systems, just keeps increasing? It's a tough question, isn't it?